Welcome to the Antibodies Online webinar about the effect of different SARS-CoV-2 variants on COVID-19 assays and vaccines. I'm going to talk about the SARS-CoV-2 proteins and in particular the S protein and its significance in context with existing vaccines. Subsequently, we will be having a closer look at the S protein structure and the effects of some of the known mutations. Lastly, I will provide some background on one exemplary S protein antibody. SARS-CoV-2, the causative agent of COVID-19, has a positive sense single-strand RNA genome with a size of almost 30 kb. This genome encodes a total of 26 proteins. 16 of these proteins are so-called non-structural proteins. That means they do not contribute to the structure of the assembled virion. Their function is primarily replication and production of new virus once SARS-CoV-2 enters a host cell. Among these are the two proteases, PL-Pro and the main protease C3L-Pro, the RNA-dependent polymerase, and the exonuclease for proofreading during genome replication, the main reason for the comparatively low mutation frequency of SARS-CoV-2. In addition, the SARS-CoV-2 genome contains sequences for 9 accessory factors whose function in virus replication or packaging remains largely unclear. Lastly, the genome contains 4 structural proteins, the spike, envelope, membrane, and nucleocapsid proteins that make up the assembled virion. I will be talking about the spike protein or S protein marked here in orange. Why the focus on the S protein? Here's a list of SARS-CoV-2 vaccines that have been approved until now for use in humans. They rely on different technologies. The BioNTech, Pfizer, and Moderna vaccines build on an RNA encoding a viral protein packaged in lipid particles. Other vaccines, like those from AstraZeneca, the Sputnik V vaccine, Johnson & Johnson's Janssen, and the CanSio Biologics vaccine, rely on delivery of viral S protein DNA and subsequent production of the antigen by infected cells. While the delivery of the genetic information for the antigen varies between these vaccines, they all rely solely on the S protein to mount an immune response against SARS-CoV-2. The two vaccines shown in the last 2 rows of the table, the Sinovac Biotech vaccine and the Indian Covaxin, use inactivated SARS-CoV-2 virions. Therefore, they display also the other structural proteins as opposed to only the S protein. However, the S protein appears to be the most immunogenic of the SARS-CoV-2 structural proteins, and one can assume that even in these whole virion vaccines, the S protein plays a crucial role. In short, the S protein is very important for all SARS-CoV-2 vaccines, independent of the selected platform. In order to better understand the body's interaction with SARS-CoV-2, it is helpful to have a closer look at the S protein structure. The S protein protrudes from the spherical virion, shown here on the left, and gives it its characteristic appearance that has earned the group of coronaviruses its name. Zooming in onto the S protein, here on the right, shows that it is actually a homotrimeric complex consisting of 3 monomers, shown here in yellow, gray, and green. The lower S2 section of the complex contains the trimerization interface and is important for membrane fusion and virus entry. The upper S1 section contains a C-terminal domain and the receptor binding domain via which the virus contacts the ACE2 receptor on host cells. While the CTD has been shown to contain many immunodominant regions eliciting a T-cell response, the RBD is the primary target for host cell antibodies. Therefore, mutations within the RBD are highly relevant for the effectiveness of antibodies, either in the host immune response or in immunoassays. Having a closer look at S protein mutations present in SARS-CoV-2 variants of concern and variants of interest revealed several amino acid changes that can potentially affect binding of antibodies recognizing the canonical RBD sequence. Especially the mutation in E484K stands out because an acidic, negatively charged aspartate has been changed to a basic, positively charged lysine. Biochemically, this is a significant change, and this is consistent with the observation that the 2 variants containing this mutation, B1351 and P1, reduce the effectiveness of treatment with serum from convalescent patients and some of the existing vaccines. It is also to be expected that some of these mutations might negatively influence the function of some existing antibodies used for SARS-CoV-2 lateral flow assays, ELISAs, and other immunoassays. In order to verify the usability of any antibodies targeting the S protein, one has therefore to test if they retain the ability to bind their target in novel virus lineages. One aspect of immunity upon vaccination or infection with SARS-CoV-2 is the production of neutralizing antibodies. Simply put, neutralizing antibodies get in the way of the S protein's interaction with the ACE2 receptor on the surface of the host cell. The main target for neutralizing antibodies is, as I have mentioned before, the S protein RBD, shown here separately on the right. Depending on the location of their targeted epitopes, neutralizing antibodies are classified into classes 1, 2, and 3. An antibody attached to these areas of the RBD interferes with binding to the ACE2 host cell receptor. Of particular interest are the class 2 antibodies, indicated in yellow. These antibodies effectively block the receptor binding motif, or RBM. This motif consists of amino acids that form contacts with residues on ACE2. Here one finds, for example, the residue 484, and it has been shown that the E484K mutation effectively abolishes binding of class 2 neutralizing antibodies, thus negating their neutralizing effect. In contrast to class 1, 2, and 3 antibodies, there is a fourth class of anti-S protein RBD antibodies, indicated here in red. These antibodies are characterized by the fact that they bind in none of the 3 sites of the other 3 classes. They do also not contact any residues that bind directly to ACE2. One of these antibodies is CR3022. CR3022 was originally isolated from the serum of a convalescent SARS-CoV patient about 15 years ago. The antibody on its own shows only weak neutralizing effect, since it binds to a conserved epitope that does not interfere with ACE2 binding. It does, however, synergize well with other neutralizing antibodies. That means the combined effect of the 2 neutralizing antibodies is larger than would be expected. Besides its function as a neutralizing antibody, CR3022 is frequently used as a reference in immunoassays targeting the SARS-CoV-2 S protein. Therefore, we were interested to see whether this antibody will bind to the S protein of the lineages B117, B1351, and P1. The antibody's epitopes within the S protein RBD do not include any of the amino acids mutated in these variants. Unless the quaternary structure of the S protein trimer has any effects on binding of CR3022, we expected that the antibody still binds to all 3 variants based on the protein's secondary structure. In order to test this hypothesis, we teamed up with Nanotemper. Nanotemper offers benchtop instruments for microscale thermophoresis measurements to quantitatively analyze binding of 2 partner molecules or complexes, in this case antibody CR3022 and different SARS-CoV-2 S protein variants. Trimeric S proteins from the SARS-CoV-2 wild type and variants B117, B1351, and P1 were diluted to concentrations ranging from 7.6 picomolar to 250 nanomolar. The fluorescently labeled antibody was added to a final concentration of 2.5 nanomolar in all of these reactions. Tracking the change in fluorescence in each sample makes it then possible to determine the dissociation constant for each S protein variant of CR3022. The graph shown here on the left depicts the change in fluorescence, plotted on the y-axis, as a function of the S protein concentration. One can see that the slope of the curve for the canonical S protein in orange and the B117 S protein in mint increase at lower concentration than for the P1 variant and even more so the B1351 variant. Since these fluorescence values are absolute, it is difficult to compare them by eye across the different experiments. Normalizing the graphs to depict relative values for the fraction of S protein bound to the antibody, shown here on the left, makes the difference very clear. The shift to the right, that means higher concentrations of the S protein ligand, becomes very clear in this representation of the data. It is also reflected in the dissociation constants for the antibody S protein binding. Binding of CR3022 to the canonical S protein has a Kd of 1.09 nanomolar. As a reference, an antibody with a Kd in the low nanomolar range is generally considered a high affinity antibody. Many commercial antibodies have Kds in the micromolar range, that means 3 orders of magnitude higher. The Kd for the B117 S protein is virtually unchanged, whereas the S protein from the SARS-CoV-2 variant P1 is already bound with a 3 times higher Kd. Lastly, the Kd for binding of the B1351 S protein is 1 entire order of magnitude higher than that for the wild-type S protein. So even though the CR3022 RBD epitopes are not affected by any of the mutations in B1351, based on the S protein's primary structure, the antibody's affinity to this variant is reduced, maybe due to steric hindrance elsewhere. That being said, according to these measurements, it is apparent that CR3022 binds tightly to all 4 tested S proteins. The wild type, as well as those of variants B117, P1, and B1351. In comparison, the Kd for binding of the wild-type S protein to its host protein target, the ACE2 receptor, has been determined to be around 35 nanomolar. So CR3022 remains therefore a valuable reference for ELISAs, lateral flow assays, and neutralization assays. A quick comparison to some real-world data shows that the effect of the S protein mutations, in particular in the B1351 variant, is indeed measurable. In this first study from Israel, up here, the authors came to the conclusion that the variant of concern B1351 does indeed escape neutralization by class 1 and class 2 antibodies. In the second publication from South Africa, the BioNTech Pfizer mRNA vaccine was shown to efficiently neutralize the SARS-CoV-2 wild type as well as the B117 variant. In comparison, neutralization was reduced almost sevenfold with the B1351 variant. So depending on antibodies, mutations in the S protein can really have significant effects. In summary, based on MST measurements, the neutralizing antibody CR3022 recognizes S protein from SARS-CoV-2 variants B117, B1351, and P1. This means that although the binding affinity of SARS-CoV-2 antibodies to the antigens may be affected by mutations, they can remain valuable tools also for novel virus variants, depending on the epitopes they bind to. We are now working on extending these binding measurements to S proteins from other SARS-CoV-2 variants, such as, for example, the B1617 variant. Besides, we are already testing additional monoclonal and polyclonal antibodies, as well as different antibody platforms in comparison. More generally, we at Antibodies Online are constantly extending our offering of SARS-CoV-2 related antibodies, proteins, and assay kits. Thank you very much for your interest and your attention. In case of any questions, please don't hesitate to get in touch with us either by email or by phone.